Biology. I'm Rohit Kumar, Biology faculty, and today we explore Chapter 5, the fundamental unit of life, cell structure and function, learning objectives. By the end of this chapter, students will be able to understand the structure and function of a cell, explain the differences between plant and animal cells, identify and describe the major cell organelles and their functions, describe membrane transport mechanisms such as osmosis and diffusion. 1. Introduction to the cell. Introduction. Life begins with cells. Every organism, from the simplest bacterium to complex humans, is made up of one or more cells. The chapter introduces the cell as the basic unit of life, laying the groundwork for cellular biology. Discovery of the cell. Robert Hooke, 1665, coined the term cell after observing cork tissue under a microscope. Anton van Leeuwenhoek first observed live, single-celled organisms, bacteria, protozoa. What are living organisms made up of? All living organisms are made up of cells, which are the basic structural, functional, and biological units of life. Historical background. Robert Hooke, 1665, discovered cells in cork slices using a primitive microscope. Anton van Leeuwenhoek, first to observe live cells. Schleiden and Schwann, 1838-1839, proposed the cell theory. Rudolf Virchow, 1855, added omnis cellula acellula. All cells arise from pre-existing cells. 2. Types of organisms. A. Unicellular organisms. Consist of a single cell that performs all life processes, e.g., amoeba, paramecium, chlamydomonas. B. Multicellular organisms consist of many cells organized into tissues, organs, and systems, e.g., humans, trees. Cell theory, proposed by Schleiden, plants, and Schwann, animals. Refined by Rudolf Virchow, who stated, omnis cellula e cellula. All cells come from pre-existing cells. Cell theory, modern version. 1. All living organisms are composed of one or more cells. 2. The cell is the basic unit of life in all living things. 3. All cells arise from pre-existing cells. 1. Structural organization of the cell. 1.1. Components of the cell. The cell comprises three major parts. 1. Plasma membrane, cell membrane. 2. Cytoplasm. 3. Nucleus. 2. Plasma membrane, the living boundary structure. Phospholipid bilayer with embedded proteins. Flexible, semi-permeable, and dynamic. Functions. Regulates entry and exit of substances. Maintains homeostasis within the cell. Transport mechanisms. Diffusion. Movement of molecules from high to low concentration. Osmosis. Movement of water molecules from a region of low solute concentration to high solute concentration through a semi-permeable membrane. Types of osmosis. Hypotonic solution. Water enters the cell. Cell swells. It is endoosmosis. Hypertonic solution. Water leaves the cell. Cell shrinks. It is exoosmosis. Isotonic solution. No net movement of water. Osmosis. Examples. Hypotonic solution. Enters cell, it swells. Turgid. Hypertonic solution. Leaves cell and it shrinks. Plasmolysis. Isotonic solution. No movement of water and no change takes place. 3. Cell wall. The protective shield. Plants only. Composition. Mainly cellulose. Non-living. but crucial for protection, support, and shape. Allows materials to pass freely, not selectively permeable. 4. Cytoplasm. The cellular matrix, jelly-like fluid, cytosol, inside the cell membrane. Contains organelles suspended in it. Site of metabolic activities. 5. The nucleus, brain of the cell structure, enclosed by a double nuclear membrane with nuclear pores. It contains A. Nucleoplasm, B. Chromatin, uncoiled DNA, condenses into chromosomes. C. Nucleolus, synthesizes ribosomal RNA, rRNA. Functions: controls cellular activities, growth, metabolism, protein synthesis, stores genetic information, regulates cell division. Note: in prokaryotes, the nucleus is absent. DNA floats freely in the cytoplasm, nucleoid region. Cell organelles, specialized subunits. 6.1 Endoplasmic reticulum, ER, rough ER, RER. Has ribosomes, synthesizes proteins. Smooth ER, SER. Synthesizes lipids, detoxifies chemicals. 6.2 Golgi apparatus, Golgi complex. Stack of flattened sacs, cisternae. Functions: modifies, packages, and dispatches proteins and lipids. 6.3 Lysosomes, single membrane organelles containing digestive enzymes. Function: break down waste, worn-out organelles, pathogens. Also involved in autolysis, self-digestion in dying cells. 6.4 Mitochondria, double membrane, with inner folds called cristae. Site of cellular respiration. Produces ATP, energy currency of the cell. Has its own DNA and ribosomes capable of self-replication. 6.5 Plastids, plant cells only. Types: chloroplasts. Green, contain chlorophyll, site of photosynthesis. Chromoplasts, colored plastids, red, yellow, orange pigments. Leucoplasts, colorless, store starch, proteins, lipids. 6.6 Vacuoles, membrane-bound fluid-filled sacs. Plant cells, large central vacuole, stores cell sap. Animal cells, smaller, more numerous. 7. Prokaryotic versus eukaryotic cells. Feature prokaryotic cell. Eukaryotic cell. Nucleus is absent, nucleoid region, in prokaryotic cell. And it is present, true nucleus, in eukaryotic cell. DNA is circular in prokaryotic cell and it is linear, within nucleus in eukaryotic cell. No double membrane-bound organelles. Present in prokaryotic cell, whereas it is present in eukaryotic cell. Cell size of prokaryotic cell is 1 to 10 and that of eukaryotic cell is 10-100 Examples of prokaryotic cell are bacteria, archaea, and examples of eukaryotic cell are plants, animals, fungi, protists. 8. Plant versus animal cell differences. Cell wall, present in plant cell and absent in animal cell. Plastids present in plant cell and absent in animal cell. Vacuoles is large central vacuole in plant cell and small, scattered in animal cell. Centrioles is absent in plant cell and present in animal cell. Shape is rectangular, rigid of plant cell and round, irregular of animal cell. Important concepts and keywords. Cell theory, osmosis and plasmolysis, chromosomes and DNA, organelle functions, structural differences among cell key takeaways. The cell is the fundamental building block of life. Organelles perform specialized functions to maintain cell viability. Differences in structure in organelles define prokaryotes, eukaryotes, and plant animal cells. Membrane transport is vital for homeostasis.